Okay, well, I think we can get going. Welcome everyone. So we've made our way all the way up to lecture 4. And it's been a ride, for me. Hopefully it's been a ride for you. The journey we're going on, okay, the journey, we're seeing this every week, particularly applies to actually what we've done so far. It'll still apply in the later ones, but this picture I have on the right here, this is the progression up through spatial scales and to some extent temporal scales in neuroscience. We've been kind of gradually climbing up from neurons to we covered like both neural mass models and also kind of abstract neural network models, Hopfield networks and so on. They're like networks of neurons. We're working our way up, and we're somewhere around here now. Okay, so neural fields is thinking about neural tissue, particularly in its spatial element, and particularly like the kind of local spatial structure of certain kinds of neural systems. So this starts to be relevant when we get up to the kind of tens of millimeters to centimeters scale. Okay, so I'm saying patches, regions. There's little snippets here. These are kind of zoom ins on this MRI image that I've got on the right. So I'm kind of zooming in, showing essentially kind of a little patch of tissue around, like within a particular brain region, essentially. If you really zoom in, you'd see that the little white dots here, those are all little triangles. So it's a kind of triangulated mesh. And these kind of high resolution triangulated meshes, they actually work quite well for thinking about neural fields at the level of like MRIs. I mean, fine points on MRIs. We won't actually cover that today, but maybe I'll bring it up in the next lecture. I think that would be a good idea. So in any case, working our way up through the spatial scales, we're up to a mesoscale aspect of neural organization, what we call neural fields. The last time we did a deep dive on Wilson-Cowan model. And Wilson-Cowan model is all about EI interactions, right? It's all about this excitatory and inhibitory neuron. I think I had the picture on the previous slide. Yeah, there we go. Like just the E and the I here, the red guy and the blue guy, doing a little dance, feeding back off each other, excuse me, and generating oscillations and things like that. And as I said, Wilson-Cowan's kind of archetypal mean field model, but insofar as we could analyze it on the phase plane, it's somewhat similar to some other two-dimensional computational models studied in neuroscience. And phase plane dynamical systems, that's all kind of a little bit on bread and butter, but it's the kind of things we see when we're looking at the temporal dynamics of those particular kind of neural models. So a lot of what applied previously continues to apply as we move along. So we're moving on to neural fields now, but the real emphasis here now, it's not time, it's space, but it's also space-time, essentially. So we'll see things like activity kind of spreading, activity like evolving over some area of tissue. But the emphasis is really less on the temporal aspect than the spatial aspect, at least insofar in the things that we're talking about. But if we go further, which we will do, but not too much today, then these things kind of mix in. So the things we've thought about with oscillations and so on, they have a role in understanding neural fields. But today we're kind of mainly going to be thinking about some modest aspects of the spatial piece. Okay, so that's a summary. Let's work our way up to there. This little movie progression here. This is supposed to give you just some of the intuition about contiguity, kind of moving towards we call continuum field models, right? So we have a bunch of points in space, and if we kind of increase the space or increase the resolution, so decrease the distance between the points, then at some point we just have a kind of continuous medium. We don't have points anymore. We just have a continuous space, and there's the key thing about the space is something about the local connectivity structure. So these type of systems, like this is not something that's specific to neuroscience, right? So it's kind of very common type of structure in terms of physics. You know, you have some compounds, some gas, some fluid. There's nothing special about here versus here. It's kind of there's a local relationship between all points in space. So there's a very strong relationship and kind of motivation in this particular part of computational neuroscience called neural field theory, like inspired by other areas of physics that deal with sort of kind of fluid-like structures in a nutshell. This kicked off, it's quite old. I think I've said this a few times already, and I mentioned kind of the history of the particularly mean field modeling approaches in mathematical computational neuroscience. They go back a long way. When we talk about neural fields specifically, I remember last week, 2 weeks ago, we talked about Wilson-Cowan equations. Wilson-Cowan equations, specifically, it's these two papers from these guys, Wilson and Cowan, okay, was in the 70s. With neural fields, we also usually go to essentially the same time. This is 1973, I think, 1972. This very celebrated scientist, Amari, and also Wilson and Cowan. So the second Wilson and Cowan paper, Wilson-Cowan 1973, is a neural field model version of the 1972 one with point processes giving us these EI interactions. But this is like the time when I think the framework and some of the mathematical conceptual elements really crystallized in something that we would call neural field. Nowadays, we're comfortable calling neural field theory. I don't think there was really a kind of concept of a thing called neural field theory as such back then, but certainly people did talk about fields. And they have been on it for a while. So, you know, these kind of citations, as Burl was at this in the 50s. I didn't realize, actually, when I was looking at these slides, I'm going to go and check this. There's this citation of Wiener, which I believe is going to be Norbert Wiener. So if Norbert Wiener was during neural field theory in the 1940s, that's news to me, but it sounds that's kind of an interesting thing for me. But some of these other ones, Burl, Griffiths, different, no relationship. And Wilson-Cowan, as I mentioned. So the ideas, if you go and read these papers as well, you get kind of similar flavor. Like this, talking about kind of, you have some pulse propagating along typically it's just an excitatory medium. Eventually people started adding in inhibition, and that has a lot of other things that enable stability. But the general question of, I've got some bit of brain and then activity here, and then what happens? How does it spread? Like that people have been thinking about that for pretty much as long as they've been thinking about anything with any level of mathematical sophistication in computational neuroscience. But Amari is one of his contributions, which will be something we think about, is a certain type of spatial kernel in the neural field called I guess we call it a wizard hat, a center surround inhibition. It's called a Mexican hat, but I think that's like a little bit politically incorrect these days. Question?
Yeah. Was that the Wiener, not sure, as in like you know, it's the same guy, like the Wiener process, kind of like stochastic. Okay, that's really interesting, because maybe he could like maybe he was thinking about representing the neural fields in sort of stochastic process.
Yeah.
That's pretty cool.
I think so. I mean, I think Wiener had his fingers in all her pies, scientifically. throughout his career, but stochastic processes is one of the ones. Yeah.
Process.
Yeah, but yeah, I mean, other things, control theory, quantum physics. I think I mentioned last week as well. So there's this. I will drop the links. I'm now mentioning it twice, but there's a lecture from Jack Cowan in 2016 where he's kind of giving a one-off talk for someone else's lecture series about the Wilson-Cowan model, and he says, well, given that I'm here, my colleague asked me to do the lecture on Wilson-Cowan because I'm Cowan. But the first 20 minutes or so, he's kind of just rambling a little bit about what he was up to when he was a grad student in the 50s. It's quite interesting. But part of it is I was hanging out and chatting with Norbert Wiener and I think Feynman and some other MIT physicists and so on. So bit of a side note there, but I guess to do for me is to check this. I mean, I'm interested whether this is just a general point about stochastic kind of continuous processes, or if there's some part of like neural theory in there. Now, well, let's put some meat on these descriptions that we're giving. So, so Wilson-Cowan kind of gave us a framing about thinking about neural systems as kind of at a mesoscale as giving us pattern formation and kind of operating, there being a kind of important level of spatial organization that is kind of one level above that. I was just thinking about individual cells and so on. So that's one part of the motivation for framing these ideas about neural fields. Another part, which is very easy to convince people of, especially experimental neuroscientists, because they see it all the time, is things like propagating waves. Okay, so this is just a really common experimental observation. It's quite hard to nailing it down with like very precise spatial detail becomes quite complicated because the brain is kind of folded over on itself in various ways, and there are as I essentially implied from my initial diagram, like there's wave-like activity at multiple spatial scales in the brain, but nevertheless, like you can see propagating waves all over the place. But they're kind of different. Sometimes the speed that they propagate at depends on the spatial scale. It isn't directly, sometimes it's directly related to the speed of action potential propagation. Sometimes it's not. Sometimes it's some kind of surprisingly slower than you would expect, given how fast we think action potentials travel along axons. But just at the level of like motivation, traveling waves is something we see everywhere, kind of if we have an idea of what we're looking at. So what they're often represented as, I've lost my, here we go. Okay. What we're often represented as is something like this, where we kind of look down on a spatial coordinate, like just call it space here. Okay, so if you just have one axis called space, that means more or less, well, it can mean one of two things. It can mean I'm just looking along this direction, like front to back, left to right, of whatever it is. Maybe it's in the brain, maybe it's just like some axis of this patch of tissue that I've got in a dish right now. Or it could be like the orientation of a cortical column or slice, which is kind of what's being represented here. So that's give at least some meaningful spatial structure to the axes. So I think we're going like so. Or they might just say it might be distance from a point. But distance from a point can also just be a one-dimensional axis. In any case, well, there's some one-dimensional axis, and then we see something that kind of goes, and then the second one's with time, right? So time versus space, and then we're looking for some angled bright spot if we're going to show as a heat map, which is a slightly more elegant way than showing it as a bunch of traces, which is what's going on here. We're seeing something we've got kind of activity essentially here, and then it kind of goes high, but then drops off, and then the next point in space goes high, drops off, goes high, drops off, and then so at some point, this point in space is active, but this point is not. Okay, so this is a traveling wave in a general sense of a kind of traveling activity fronts would be maybe a more technically accurate description of that type of thing. Okay, so thinking a little bit about neurons and using a bit of the nomenclature and diagrams that we've been building up so far. So we're talking about neurons, talking about neural activity, but we're being quite general intentionally here. So we're going to be again operating at a meso scale where really we want to say something along the lines of on or off or high or low. And the thing that is going to be on or off or high or low, you can think about as a firing rate or an activity level of some cells, which would be, typically we think about that as being at the soma rather than at the dendrites here, but it's at some point in space. So we've got activity, just calling U here, okay, and then we have some other point in space. I was going to say U Y. So we have 2 points in space, X and Y. What we're gonna say is that the activity at X is at a certain time is going to be a spatiotemporal convolution where we have this guy F, which is our temporal firing rate function. Okay, so we've seen this before, giving us like a alpha function, kind of exponential rise and decay over time for some point input. So we have activity over space. We basically have a convolution over space over time, and a convolution over space. Okay, so thinking about our activation function, which I had before. We were at some point, I think in the first 2 lectures, talking about this kind of FI curve, firing rates, a function of inputs. This would be a typical way of thinking about activation curves at the level of individual neurons. What we also moved to, there are two other types of activation curve that we thought about over the last few lectures. One is where this is rather than a kind of baseline and then a kick up, and then we have some mostly linear domain, just increasing linearly after a certain threshold point. We also thought about sigmoids, where we have something smooth, still kind of saturating at 0 and then going up and then saturating somewhere at the top, which this one doesn't do. And we just talked about heaviside functions, so just off and then on, so kind of delta function on at a certain threshold point. Each of those are all valid ways of understanding, describing the ways in which either a neuron or a bit kind of part of neural tissue responds to its inputs over time. We're mainly going to be thinking about the heaviside case today because it's simpler, but everything can be kind of fleshed out with more temporally rich activation functions when we want to get there with a little bit more detail. So this is the kind of general setup. We've got activity at space and time. Going to be some spatiotemporal convolution of activity somewhere else at space and time. Okay, exponential synaptic filter. So then for this, there's a integer differential equation corresponding to this convolution expression. Going to be telling us how activity evolves over time, just in one dimension. So we have just one spatial dimension x, and the main thing that's interesting here that we're going to mainly talk about is this integral over space. Okay, so dy wxy. Okay, so this structure, this general setup, is basically like, it's exactly the same. It's the same as the basic components of our Wilson-Cowan equation. If we can drop the EI bit, just think about E, for example, and drop the self-feedback on E. So that was this. This was like the first half of Wilson-Cowan equation before we had like plus I and plus self-feedback on E, where E would be U. So this is, so in other words, like the neural field equation is basically the same as Wilson-Cowan equation if we drop some of the extra bits of the Wilson-Cowan equation that gave us the oscillations. The motivation for this beyond, so I talked about traveling waves, so things we see as kind of activity fronts. There's also an anatomical rationale for thinking about things as continuous, spatially continuous and contiguous anatomical structures. So this is kind of summarized a little bit by this sketch here, this diagram here from Gustavo Deco. It's highlighting, it's kind of showing two things here. So this is like the parenchyma. This is the superficial white matter, and this is tracing of an axon with a fluorescent tracer. Oops, excuse me. So there's two things going on here. One is at each point, so let's say A and B. It's not emphasized so much in here, but if we kind of look inside these squares, we've got some, and take that point as being our kind of point UX. Then there's some local connections there. Let's say the local connections are like the green stuff you see inside there. Okay. And in addition, so we have local stuff, we have long range stuff. Now, the question, the point that actually these guys are making, that I don't want to get to, is there's two ways of dealing with long range stuff like this. One is to assume, which is not a bad assumption, that that's one part of a continuous distribution of connections of axons, essentially. Let's say in B, so let's say there's like a lot of dense local connections for B, and then there's a small number of long range connections. And actually, there's a good number of ones all the way points all the way in between. So that'll be a continuous, spatially continuous connectivity structure. The alternative view that these guys are making, that we're not actually going to go into this lecture, but we'll kind of build up to in the second part of the course, is where this long range connection is kind of an exception to the neural field structure. It's kind of an add-on. So we have a two-level structure. We have some spatially consistent but exponentially decaying connectivity structure locally, and then we have some long-range connections that are stronger than the long-range connections coming from the spatially continuous kernel. So that's kind of two-level neural fields, but that's the main thing that we want to think about for today is the simple part of that, where we think about some point in space connected to other points in space with some like spatial decay. And come back to Amari. So one of there's several things, but one of the new ideas that was introduced, or framings, let's say, was introduced by him in that 1973 paper, is a particular type of connectivity, or the analysis of a particular type of connectivity kernel. So this is space. Okay, it's actually x minus y, not absolute x minus y. It would be just x minus y. So a typo there. And basically we can, so there's a feature of what we call the lateral connectivity structure at the, let's say, roughly like millimeter spatial scale in particularly the cortex, but I think it's the case in other parts of the, like the retina, for example, where we have this thing called center surround inhibition. So, and then this is actually the related to the retina and kind of visual system, retinal ganglion cells. So you have center, the plus is here saying for some point in the middle, we have, and then this is going out in kind of X and Y, so two-dimensionally. We have some local excitation, and then you move out, so we get so then we have a domain of inhibition. And there are actually cells that have that, and they have the opposite, and these are considered to have important kind of computational functions from the point of view of the visual system. It's kind of quite clear, I think, intuitive how to think about it. It's along the lines of a kind of foveal spatial attention, if you like, where you say, okay, I'm kind of looking over here. I'm gonna assume that maybe stuff nearby where I'm looking is relevant because maybe I've got a bit of error, or I just want to have a certain amount of field of view. But outside of that, I want to start ignoring things because they're getting in the way. So center surround inhibition at a kind of cognitive level, like abstract cognitive level, has that kind of spatial attention kind of function just kind of naturally built in, but it doesn't need to be visual spatial in that kind of concrete way. It could be kind of just at the level of, I've got some neurons over here, some neurons over here, and the neurons or the neural tissue over here is, say, doing one thing, neural tissue over here is doing another thing, and the neural tissue kind of next door to one of these is doing something slightly similar, but it's not, but not similar enough. So I want to suppress it. Okay, so there's things outside of just thinking about space and spatial attention where I think this kind of heuristic of center surround inhibition is like clearly has a functional role. And in terms of how that fits into the neural fields approach, it's a particular equation for a synaptic connectivity kernel. So a spatial synaptic connectivity kernel, which is the weights of our network, if you like, the weights of our spatial kernel, but that it kind of connects the points in space, in this case, one-dimensional space, and the kind of structure of this, that we're basically thinking about two pieces to it. They're both exponentials, but this guy here is just a difference of exponentials, just some exponential decay. The difference between them is these parameters, these kind of subindices for the variance, the width, and then the difference between them gives you this center surround structure. And then when you kind of incorporate that into our models, then there's a few kind of dynamical phenomena, spatiotemporal dynamical phenomena that can come out of having a center surround inhibition type structure. But yeah, difference of exponentials is the way we think about proceduralizing that mathematically. We're not going to do much with this per se, other than it will appear once or twice in the next few slides. So it will come back, but the specific structure of the equation isn't super important. I'll just think about it as an exponential decay over space. So there's three things that we want to be thinking about from the point of view of analyzing neural fields and neural field equations that we're going to go through here. Question. Hi, Professor. Could you repeat as to why we take difference in spatial coordinates? Are we looking for average behavior in state space? What does the kernel tell us practically? Yeah. Okay, so I think Ravindu, you're asking about the x-axis here, x minus y.
Yeah, that's right.
Yeah. So the point is that it's a translationally invariant kernel like structure of a space, right? So at some given point, we have this kind of this drop off, but we can see the same thing at another point. So the actual value of x and y doesn't matter. So from the point of view of describing the kernel, we just need to say it's a function of the difference between x and y, or the kind of extent, if you like, the distance that you go away from a central point. So in 1D, just the distance you go back and the distance you go forward, but it doesn't matter what the value of the center point is. What we're talking about here is just how the kernel changes as a function of the distance from it. So that's why we've got a difference here. And yeah, the general answer is, because this comes up again a few times, like why differences, it's because that just allows us to do things like do certain difference operations that aren't dependent on specific points in space. And just more generally think about translationally invariant connectivity structure rather than a particular spatial pattern within the connectivity structure. What does it tell us practically? Well, I mean, I think there's two types of answer to that. One is just what does it mean anatomically? And I tried to give you that before, but I probably could do a better job, or we could say more about it. But in a nutshell, like you have some neurons here, they're really densely connected to the ones near them, and they're less densely connected to ones further away. And then in addition, there's this excitation inhibition locally versus slightly further away aspect to it. So those are practical points about the spatial structure of the synaptic connectivity at a kind of microanatomical level. And then the other part of the practical points is like what does that mean for their activity? That's like the point. That's what we're going to be looking at. Hopefully that's a start on answering that. And then Krish is saying, it is the symbol, yeah, that thing. Yep, that's a convolution. That's exactly right. So we have a spatial convolution, and we have a temporal one, because we also have activity. We're also looking at activity over time. Okay, so there's a kind of spatio-temporal convolution that we're going to turn into an integral differential equation. Okay, so there's three things that we want to do as a start when we're thinking about neural fields. We want to, there's three scenarios that are possible when we have. I'm going to just try and set this up. What's the best diagram to set this up? So, I mean, this one. I want to say, yeah, I'll just use this one on the top right here. So a lot of everything we're going to talk about for the rest of this lecture, for the next half an hour, is going to be one-dimensional in space. there's several ways you can motivate one-dimensional in space from a neuroscience point of view. However, for the most part, the kind of structures that are relevant here in the brain are either 2 or 3-dimensional in space. In reality, let's say they're 3-dimensional insofar as if we think about laminar depth, we're thinking about the cortex at least. It's a bit different with subcortical structures that don't have a kind of sheet based structure. But if we just take the cortex for sake of argument, the cortex is it's like the surface of a ball or some other spheroidal object, if you like, that has a little bit of depth. So the kind of leather of the ball, let's say, would be your kind of 3rd dimension, your Z dimension, let's say. And then the kind of movement along the surface of the ball would be a kind of X and a Y direction. So that's actually like the best way of representing the organization of the cortex. It can so the field structure there can be, you can think about it as 3-dimensional if you include the Z axis, which is basically going down a cortical column. But we can also, and we usually do, to be honest, the kind of what I do, we just drop the Z dimension because it's really shallow. So it's not very, so there isn't like a whole lot going on in terms of, now people who do laminar anything will take issue with that, but at a kind of mesoscale, we can just drop Z and then we just have 2 dimensions in space. But for a lot of the analysis we want to do, and including the actual like things that look like brain activity, not just say toy models that are easy to analyze, we can just drop the 2nd dimension in space and just thinking about 1 dimension of space. So that's what we're kind of doing here. We've got 1 dimension of space, kind of called X. So you can either think about this as, say, I've got say a part of the visual cortex that's here and another part of the visual cortex that's a little bit further up the head, right? And we're just kind of tracking a line in some Euclidean coordinate. There are you can use other heuristics if you like, but it doesn't really matter. We've got some spatial dimension X. And then we're going to be thinking about activity. And we're going to think about what are kind of relationships between space and time, given the thing that we've talked about a little bit and we'll continue talking about, which is the spatial like connectivity structure along this x-axis, and the way in which the dynamics of the activity are kind of driven by the spatial connectivity structure, right? So it's going to be space X and time Y for most of the things we're talking about. Thank you, Parser. Shivank, hi. In the plot, why does the function transition smoothly instead of abruptly like the hempers? Good question, Shivank. I skipped a slide. I mean, you'll have noticed like I am going to go back. Yeah, sure. Well, I'm going to go back and we can just use this one. So same thing here, okay? So in this case, it's U rather than X. Well, sorry, U is the variable. X would be like points in space. So it's still the same kind of thing. And yeah, so we consider like this is something that's more like distance from a point now rather than some actual coordinate on the x-axis. So in principle, this could be representing a 2-dimensional structure or a 3-dimensional structure if we just like define the dimension as being distance. In any case, yes, I think we'll also, why does it transition smoothly instead of abruptly? That's something that we'll see. The answer will come in the next 1 or 2 slides. Shivank, so pay attention. Okay. So we're going to be thinking about activity over space, but also evolving over time. And in one dimension of space. In this scenario, also in more dimensions of space, there's basically three scenarios that we're that are possible and that are analyzable. So we call them fronts, bumps, and traveling waves. So a front, I'll just describe them and then we'll kind of work through and we'll take them in corners. So a front would be, it would look like this Heaviside function, but I don't need to confuse it with the Heaviside function. Yeah, so I've actually, okay. Let's use the next slide for now. So front will be basically where we have some regime of low and some regime of high. And we just have a split regime of low, regime of high. A bump, which we'll see in a few slides, will be when we have a kind of regime of high that on both sides drops down, as opposed to the front, where we have just one transition from low to high. And then a traveling front will be a bump that moves in space over time. So we'll see the pictures and we'll kind of get more of the intuition of that as we go. So I'm going to talk about, and these are all spatially localized solutions. Okay? So the thing we want to focus on now, so we have our general equation here, written as a convolution, as write it out as a rewrite it as integral differential equation here. So we've got evolution over time of this variable we'll call Q. And what we want to do is we want to drop time. So what we want to be able to do is say, if we set the temporal derivative here to 0, then we're going to have an equation now, just equation that's kind of non-changing over time, that's some function of, as is always the case with these systems, like some activity at some point x is a function of inputs from other points, okay, but it doesn't change over time because we've set our derivative to 0, so it's stationary. It's a stationary solution. So this is what we're after here. We're going to say what are the criteria for these kind of solutions, and how do we find them? Now, the approach, so we're going to take the front in first, and then kind of move up to bumps and traveling waves. So we're looking for something that's going to give us a solution to this equation here. The approach here is going to be, we're going to split on some threshold. We're going to look for solutions where we can define a threshold called threshold theta, for which in the domain where x is below the threshold, so x here is less than 0, sorry, so we're going to split into, we have x below 0, and we have x above 0, and we want to find a threshold for which we have activity is going to be switching from 0 off to on. Okay, and then we've got something going on at 0. That's our threshold. So connecting the previous two. So this was our Heaviside function here. We want to say for some activity value of activity u, we're going to say, so the Heaviside function here is saying we're going to have some like discrete threshold point where above that threshold point we're going to call it firing is on. Below that threshold point, we say firing is off. So flip from 0 to off to on. Okay. So what we want, the way to approach this is say we're going to just introduce a change of variable. We're going to add this guy y prime in. So we're going to substitute y prime on the right hand side, and this is going to let us get a expression that we can solve where we don't have x in on the integral for below and above 0. So this gives us this is going to give us our solution for our stationary front. In the case of the Heaviside function, as I said before, we can substitute in the other spatial kernel structures. So the kind of hazard, this is how it looks for the Mexican hat, for the exponentially decaying connectivity that I showed before. The difference being, so we're just going to get a kind of smooth variation, and with this one, kind of smoothly varying into the threshold point. And then what it's going to look like in space is the following. So we have, as I said, it's stationary in time. So we've set dt to 0, d by dt to 0, with nothing changing over time. But we have a transition where blue is off, x is, sorry, yeah, so blue is off, x is, or yellow is on, actually it doesn't matter. We have a point where, so this part of the patch of tissue is just not firing. This part of the patch of tissue is firing, okay? We just have a transition point. So this is the simplest, apart from just flat, this is the simplest kind of spatial structure that we can understand from the point of view of building our way up on neural fields. And yeah, you just think of it as like when maybe one intuition that works, or this works a little bit better for the ones we'll get to, is so we have some input or some cognitive state or whatever it is that makes neurons fire in some patch of tissue, and the organization, the kind of spatial organization of the connectivity structure of the neural field, like allows for a situation where there's one part of the tissue that's like activating, or is not just activating, but kind of maintaining its activity over time because it's not changing on t. And there's another part that's off, and they're not flipping around or any kind of temporal variation or any kind of complex pattern, right? So it's a simple type of solution for the one-dimensional field. Okay, so our second example, we're going to think about a bump. Okay, so this is just one level up from our previous one, where we just had like one transition zone, had low to high. In this case, we've got two transition zones now. We've got low to high to low, and what we've got is the domain of Q, so the domain, kind of part of space where our activity is above our threshold, is living inside an interval. An interval that's going to be kind of 0 to delta, and defined here. Okay, and so yeah, and so we have two transitions: transition into and transition out of. So basically same as what we had before. But in this case now, rather than just kind of two criteria on this, before we just had one thing for zero, a special case for zero, at least for this one. So for above and below. Above and below thresholds. So now, because we have two thresholds, we have two. Sorry, I'm going on. We have two zones. One we're crossing points. Sorry, where we're going into and out of. So we have three conditions on the right hand side that we want to do a kind of piecewise solution of. So these can be kind of solved separately, and together gives us the full solution for this case that gives us something that we'll call a bump. And then visually and conceptually, it's like this, right? So it's kind of a bump or a band, right? It's a bump over space. Space is our x here. Confusingly, we put x on a y-axis. T over time. So we have some localized domain of firing that is called sustained firing. And again, the way to think about this from the neural organization point of view is like this is some part of the patch of tissue that you're interested in that maintains its firing. I mean, in this context, like infinitely, but obviously nothing works like that. But it's like it's just this is maintaining a high firing, this maintaining a low firing, and these two kind of work together. That's our second simple type of solution. And then the third one is a traveling wave. So the approach to this is basically the same, but now we're not going to have we're not going to look for something that's fully independent of time. We're looking for something that's independent of time when we basically apply a correction or a kind of warping of time, which we call a co-moving frame. Co-moving frame is this guy here. So we'll call this psi. And so we're going to define some interval or some interval over space, multiplying on some constant. We'll call it basically is the speed. So this is kind of flipping the thing that you would see as like so. It's essentially like flipping something that kind of angled over time into something that's straight. That's one way of thinking about it, like adjusting the definition of time, or the kind of window frame definition of time, so that we're kind of aligning previous points to current points as a function of how far back they are. So that's kind of what our quantity here is. So yeah, this Q of psi. So once we kind of define that, and all we need to define that is basically a definition of a velocity. So that there's another parameter in here. Then we can find solutions that have the same structure. And what this looks like, come back to some like relevant data. So this is some experimental data and some data from a simple model, similar to what we're looking at here. Just to reiterate a point that we were seeing, I think, in the figures that I showed earlier. So we really do see this kind of activity pattern. So this is our traveling wave, or our traveling front, I should say, over time from a small slice of tissue from cortical layer 2-3 in mice, I think. Okay. and there's a pulse that's been applied, brief stimulus close to the electrode. So they observe this thing that's just easily described as a traveling front that we can understand as one of the types of stationary solutions that we can see in a one-dimensional neural field. Now, I put the slides up, and there's more to say on this, but I'm gonna skip this bit so we have enough time for our tutorial later. But they're just for people's enjoyment. There's some extra content in here about how we can study the stability of traveling waves on of these kind of traveling waves. It's using something called Evans functions, but we're not going to go into that today. The final piece that I want to add into the picture, quite briefly, really, is thinking about time delays. So this was our basic setup. Again, this is the same slide that we had before. So we were thinking that there's some input coming from other parts of our tissue structure. The input from other parts of the tissue structure is defined by the shape of the synaptic connectivity kernel. The whole thing evolves over time, but the shape isn't time. The shape doesn't understand time. So the connectivity structure doesn't understand time. That works if propagation is instantaneous. Propagation between adjacent points. But that's a bit of an idealization. Sometimes it's an appropriate idealization, sometimes it's not. But say complete, physically and physiologically complete representation of how we should understand how things are kind of working in this context is that our kernel is not a spatial kernel, it's a spatiotemporal kernel. And so where there's kind of two things going on, there's input coming from all the other points in space now, and there's also input coming from all the other points in space in the past. Okay, and then the addition there is that we have that to some extent with our convolution, but the kernel itself doesn't explicitly have time, temporal convolution. So the full picture has inputs coming in from different points over space, inputs coming in from different points over time, and the inputs from other points in time is going to be delayed by some value tau that is also a function of space. And we can even think, and it's not necessary, but it can think of it's right in the present context. We think about this as being a spatially invariant, the translationally invariant function of space, where we can just say absolute value of the distance from between these 2 points, right, between x over here and y over here. So that's the distance over speed. It's going to give us our time delay. And so there's going to be input arriving at here from here, but it's going to be delayed by some value tau, which will go here. We're going to have a retarded convolution in this case, retarded integral. Let me go over here. So now we've got just a small addition to the kind of right-hand side here, where we're integrating over our kernel, our connectivity weighting kernel, but we're also kind of moving backwards in time with our delay. So our delay there is kind of written out explicitly there. It's y over v, so velocity y rather than we had the speed, we had a c before. We've got s minus absolute value, s, yeah, so s minus, so distance over velocity there, so t minus tau, tau is s minus abs y over, it's not v, it's eta, isn't it? So this guy here. So that's our kind of time delay in the kind of, again, a more complete version of our neural field equation. All of the analysis basically can still apply, but we're just going to have an extra integral back over our time delay. But we can think about they still give us traveling wave solutions, and we can understand in the same framework. So I wanted, yeah. So if we recall, so this is our fronts, okay? So in the case where the example that we had before, where this was our kind of stationary front. So this is space, it's time. We have a switch from low to high activity that is stationary over time as we go up here. When we have the kind of physical case of a positive conduction velocity, then we have, again, we've just kind of angled and, you know, warped the activity front, but it's the same type of thing. So we've still got a stationary front, if you like, but it's stationary with an adjustment over space, accounting for the delayed input within the connectivity kernel. There's an extension of the Evans function stability analysis with the delays that I'll leave to you for extra reading. And I just want to wrap up with some examples and just connecting a few of these points to things that we had in the previous lectures. So we mentioned Wilson-Cowan already, or Wilson-Cowan type circuit. It's a Wilson-Cowan EI, self-feedback on E, self-feedback on I. So yeah, okay. So Ravindu says, for the case of time delay, which circumstances would you opt in to include the delay as opposed to not opting in? Is time delay like a more accurate representation of phenomena? It's a very good question. So we usually include delays when the distance in space exceeds by a large degree the synaptic integration time period. So if you remember, well, do I want a diagram to show you this? I'll use a diagram to make this point, but I've got to go back a little bit. I just want you know, actually, I think the one we had before, that little neuron over here. Actually, the best image I have to make the point I'm going to make is the one that we had right at the start from Deco with the fluorescent thing. Yeah, this one. Okay. So there's a fairly concrete answer to your question. Just to repeat the question, saying when should we include delays? When should we not? So I have two answers to that. One is at a kind of fundamental physical level, there's always delays should always be considered to be part of what's going on because we don't have any actual instantaneous, you know, wormholes in space or anything that should even be thought as like approximately instantaneous, like you know, light and electromagnetic stuff. That's kind of often very fast, right? So stuff is quite slow in the brain. However, basically the answer is when we're, say, thinking about inputs from inside this box to the point that's the middle of the box, which would be, let's say, like that's 0, like some point, the point in space where we're kind of collecting our inputs from. And then each of the points in space, each of the neurons, this is where I do want my little dendritic tree. Okay, I don't have it here, but so if you recall, like each of these points in space, it's going to be a bit of either a neuron or some population of neurons that are receiving some input on their dendrites. Then they have some synaptic response, and then they'll receive like a lot of that or a little bit of that. They'll have some firing threshold function. And then if they exceed their firing threshold function, then they'll fire, but that could be a continuous firing rate rather than just a kind of binary on-off. But they'll transform their inputs to their outputs, okay? Now, that is something that's not instantaneous. That happens over time. And the period of time that over which that occurs is essentially a few milliseconds. So this is the case. I mean, even for neurons, like it's more or less this, but certainly if you have a small patch of tissue, the time at which all of the neurons in that small patch of tissue, if you have some input come in, the time it takes for them to receive the inputs, neurotransmitter release, hits some receptors on the postsynaptic cell, induces transmembrane flux, action potentials, all that jazz up to a firing rate. That's something that takes a few milliseconds. So the answer to your question is if the delay, so if the kind of t minus x minus what am I looking for here? X minus y over v. Okay, so the distance over here divided by the velocity is smaller than the, say, a few milliseconds. In other words, the time it takes for these points to integrate their inputs, then we usually don't attach an explicit time delay to them because we can't really resolve the delay associated with the synaptic integration and the delay associated with the transmission over that that distance in space. However, the time when you kind of should include time delays is this guy here, right? So this A to B. So let's say that the time it takes for activity to go along this axon here from A to B is much longer than the time it takes for B to process its inputs. And to be concrete, like much longer than a few milliseconds. And to be very concrete, tens of milliseconds. Because this is like what you're looking at, basically, in terms of conduction delays in axons. If that axon is usually, say, over a few millimeters, over a few centimeters. So very long axons in the brain. There are fast and slow fibers within them, but we usually kind of ignore the slow fibers and say like the average propagation delay for a very long connection, say from the back to the front of the head or from the left to the left hemisphere to the right hemisphere, is on the order of 100 milliseconds. Okay, and that's for something that's, say, between 50 and 100 millimeters long. So in those cases, 100 milliseconds is a lot longer than 1 millisecond, right? So the synaptic integration time is a few milliseconds. If the delay is tens of milliseconds because the axon that it's moving along is quite far away, then that will be the that would be definitely the time when you should include time delays. The wrinkle to that is, how do you define what that distance threshold should be? And the wrinkle to the wrinkle is that it's kind of continuous, because you have distributions of velocities. So then you have to you kind of expand that out in terms of not a kind of discrete delay, but a delay distribution. And those then can get quite hairy mathematically, but at a conceptual level, that's what we're that's how people think about the answer to that problem. Okay, does that answer your question? Good. Okay, good to hear. So I think I was kind of done on this slide, but I was just saying that the time delayed traveling front is really a stationary traveling front. We've just applied some correction to the correction to time effectively as a function of space with our co-moving frame. And yeah, we were kind of moving over to the examples, which is what I'm going to wrap up on. But just to kind of underscore a point that I made before, I kind of started making this to say, like, the neural field the structure of the neural field we were talking about is kind of similar to the Wilson-Cowan equation. What I was telling you to ignore was like basically all of this. I mean, we can have EI neural field, but the relationship between these two is like, you think about the E and the I being kind of, or being 2 points within this kind of extended location in space, or domain of tissue. So the Wilson-Cowan equation is basically describing our point process, right? We think about it as kind of points in the brain, or like a single cortical column. We think about it as an isolated unit. We can also just unfold it in space and just treat the whole thing as a continuous relationship between populations of excitatory and inhibitory neurons that have these spatial synaptic kernels, where it's not kind of, we're not just integrating the input from the I population, and which would be like the input here, right? The inhibitory input to the excitatory population here, and the excitatory input to the excitatory population here. But these inputs are just coming from one thing that we don't have to give it a kind of a definition of space. What we also did as well is we had a networked version of this where we had, say, lots of these points. And we had some sum over a network, and we had a bunch of weights. So we kind of had a matrix of connection weights rather than a single connection weight here. So the extension here, or the point, the kind of generalization, is basically to say, you can think about this as being, or the generalization of this being, well, rather than taking the input from one point in space, I'm going to take an input from all of these points in space, and find what their kind of summed input is, and then that kind of gives us this part of the term for our like time evolution of our E and I variables. We're just going to unfold over space in a continuous way. Again, some stuff on bifurcations here, but I'm going to skip. I want to talk about some applications and like what kinds of things we see in data and in things that concepts in neuroscience where some of these neural field ideas apply. There are more than what we're going to talk about here, but these are just some examples. So one is perceptual switching. So this is I think we would call this a phenomenological model of a visual system organization where we have a one-dimensional neural field, and we say this corresponds to a kind of visual percept, but it could be like a kind of part of the visually responsive neural tissue over space. Doesn't matter too much, but the important point is the nature of the inhibition and excitation over this domain. Okay, so you see we have like excitation, inhibition, in this kind of schematic of the organization of the pathway here. Okay, so the thing that we want to, the context that we want to think about this is okay, binocular rivalry. So we actually I don't have the diagram for them showing how this works, but I guess what you can think about is what you see here. So this is what you're seeing on your left eye. This is what you're seeing on your right eye. So this is a kind of visual stimulus. So in a spatial, experimental context, it would be kind of a grating that's kind of going, the bars are going left to right on your left eye, and same thing on your right eye. A phenomenon that we're talking about here is called binocular rivalry. What that means is essentially like what you, in this experimental scenario where you have something, you have a different input going into both, into your visual system, you don't see both of them at the same time. You only see one of them, and what you see is one or the other, but varying and kind of flipping. So this is called binocular rivalry, and the way in which the kind of field structure gives us some explanation of that is that we can have, if you recall, we had like a kind of stable zone and then an unstable. We had our fronts where we have this part is on, this part is off. Okay, that's and the separation between the two is coming from the kernel structure, spatial kernel structure. So that's something that can explain the phenomenon of binocular rivalry, where basically we've got one part of our kind of perceptual system that is, say, on, and when on, we kind of corresponds to you're perceiving the thing you're seeing in your left eye, and then they switch. So this guy will be on, and you'll be seeing the gratings going this way, and then at some point you get just a switch. The reason for the switch is usually, we think of it as being a kind of fatigue kind of structure. We have some switch, and then the activity, and then what you see is then the other one that's being presented to the other part of the other part of the visual field. So this one takes over. So we have this kind of flip-flopping between input to this guy and input to this guy, that's accommodated by the fact that we can basically have one stable zone on, one activity zone on, and then another activity zone off, and the inhibition between them allows for the kind of flip-flopping. So another general, this is some of the general ideas about what we can think of the kind of computational role of the spatial part of the neural field organization that we've been thinking about is, so we had all these shapes over space. We had bumps. We had bumps that move, that we call traveling bumps or traveling waves. We had traveling fronts as well. What do we make of these? So these are just some kind of ideas, some concepts essentially to think about what are cognitive computational roles of some of these things that we've seen can come from neural field systems could be. So a common one is memory. Okay. And the simple way to think about this is that the thing that we were looking for when we were looking for our three types of stationary solution, I mean, all of them, but particularly the second one, the bump, this is activity is on, it stays on. Okay, it stays on, doesn't change over time. That was the definition of it. So I think for maybe, I'm not quite sure if it goes all the way back to Amari. Maybe it does go back to Amari. Pretty much as long as people have been thinking about these kind of features in neural field models, they've been thinking about an idea of something persisting over time, and you can think about it like activated and then persists in the absence of the activation. Okay. And this is what memory is, certainly short-term memory. So usually when we say where do we think this is going on, but if you kind of go to the kind of literature on neural field modeling in the brain, for things like localized bumps and the corresponding experimental data, it's basically the frontal lobe. And the idea is you have some kind of patch in the frontal cortex that kind of stimulus comes on. The task is to remember some feature of the stimulus. And then what you see is a period of time where neurons are firing in this particular part of the frontal lobe. They continue firing a long time after the visual part, the perceptual part of the task is finished, and they're basically like considered to be a kind of short-term memory buffer where you're retaining the information that you at some point worked its way up your perceptual pathway. And this kind of organization where you have a kind of stable patch of neurons that just is retaining its activity level, and isn't dissipating over time, that's thought of as a kind of memory function. So that's memory. Selection. So this is another kind of scenario where we might think about kind of amplification. Okay, so have some input into two different parts of space, or if you like, two locations in space. Think about them as kind of different attractors, essentially. Like, can we bias activity between this part and this part? And yeah, so kind of extension of just saying, well, we've got some persistent activity. We've got activity, but we've got a few different options. And so your different bumps, if you have a few of them, would maybe correspond to different different kind of representation options in your like representational space. And then there are other things we can kind of look at in the response to inputs and other parts of the parameters that kind of interpret in terms of selection between competing stimulus interpretations, if you like. So there are a few, yeah, there's more. Like this is the general point here is just think about what, and this is one of the features of this area of research, is we have something that's got a lot of abstraction, but it's got a certain amount of basic underlying neurophysiology, and often we want to connect it to something cognitive. So what is the mapping here? And it's often, yeah, some kind of combination of, we've got some patch of tissue that we think does something, and stuff is on or off at some points in that brain area or not. And the way in which it kind of moves between them is something we think about in terms of like memory, attention, selection, perceptual responses to different kind of inputs. So another of those is visual hallucinations. This is something that's been a popular topic in neural field models. Steve Bresloff done a lot of this, Paul Bresloff, sorry. And the motivation coming from this partly is that the type of systems that we're looking at with neural fields, particularly when you kind of go into the two-dimensional case. They're very close to like pattern forming systems, that reaction diffusion equation type systems that are studied in other fields like chemistry, where you could get kind of emergence of certain kind of spatial patterns, like Benard cells and cheetah spots and things like that. So those kind of things, they also are kind of part of the elements of typical types of visual hallucination that you can get in a number of ways. You can get these experienced from psychoactive compounds. As you just press on your eyeball, you get it. Close your eyes, dreaming. Lots of contexts where people experience visual hallucinations. They're kind of a universal thing. The general thinking, we'll just kind of describe it, is that you can kind of bias, and if we kind of look at some of the, if you go into some of the reading here, and I can point where to go. You can generate these like rich spatial patterns from neural fields. It's quite easy to do, actually. They kind of just get these jazzy movies of high and low activity levels. We'll start Zoom, folks. We're looking back. And the question is like, when does that happen and when does it not? So some of the ideas that are often discussed there is basically, so we have some, say, hallucinogenic drugs. Can we back in? Still going? Yep. I have some hallucinogenic drugs, for example. We have some kind of activity level that's being modified by some ideally transient change in the visual system or another brain system. So we can parameterize that in terms of something like the gain function, right? The activation variable, the activation function in our neural field. So that's something that what we're often looking at, or people who are studying these things are often thinking about, is like, can I kind of move a model of the say early visual cortex into a kind of certain type of pattern generating structure, not quite like this, but they often look a little bit like this, as a function of some kind of order parameter, essentially, some number that I can kind of understand with some level of physiological connection, right? As I was saying, like a kind of gain boost from noradrenergic drugs or serotonin, kind of boosting some of the gain properties of neurons, kind of moving you into a scenario where you're kind of more likely to just fall into a certain type of spatial pattern generating regime in, say, the visual system, like V1, which is where you would then perceive a kind of these kind of pattern structure in your, say, your visual field. So neural fields and hallucinations and stuff, that's kind of a long time topic. And then the final one I'm going to move through briefly is this idea of this being one of the ways to understand epileptic seizures. This is frequency time, but we also have space. So one of the things that is a kind of characteristic feature of epilepsy is you have some epileptogenic focus where tissue starts firing pathologically fast and strong. We call it a seizure focus or a seizure-like event. And then, depending on various things, of how acute the epilepsy is, if we're talking about clinically, it propagates. So epilepsy, particularly the aspect of epilepsy that you really want to deal with, which is the way in which you have some epileptogenic tissue that then kind of generates a seizure and then sends it out to the rest of the brain. And then in the acute case, you have what's called a grand mal seizure, where basically a large amount of the cortex is experiencing a seizure, that's a traveling wave of high amplitude epileptogenic firing. Travels travels locally, and also travels globally, and it's something that's very clear part of the measurement of having like in the with depth electrodes in kind of preoperatively, so you can just easily see, you can easily see it from scalp EEG as well. So the seizure focus is there, say in the hippocampus, medial temporal lobe, and then kind of propagates out to the rest of the temporal lobe, then other hemisphere, and there's this kind of spreading phenomenon. So understanding epilepsy as a kind of manifestation of a traveling wave is another of the physiological topics that's a prominent question that's studied in the context of neural field models that we've been seeing today. I'm going to wrap up. I think I had a question.
Kind of about the epilepsy slide you just had. Would the kind of field representation models also have the same kind of activation functions, and also like the same tau, like the same like speed at which the neurons fire? Like, could it be thought of as like a regular activation, regular speed, but just an overload over the whole brain, just like a huge wave everywhere? Or is it special? Like, does it have its kind of, could we model, is it better modeled with like different activation function that would insinuate like extra activation, because we're kind of seeing almost like so much activation during the seizure?
Yeah, it's a good question. I think the answer partly kind of depends on the model, and then the model depends on the theory, and then the theory depends on a lot of things like the scientist who's working on it and the data they're working with and who they're influenced by and things like that. But you know, that would lead to like the actual equations that are used in a particular, you know,
I guess
study. But
what's better asked is it common practice for the activation functions and kind of, I'm messing up the word. It's not time dilation, it's time inconsistency. Is that how you described it? What is it?
oh, the co-moving frame.
Like how time
delay. Okay.
The activation function and time delay, the ones that we looked at earlier when we were just exploring that concept, is it common practice to kind of use some of those that we looked at to model these like epileptic waves as well? Like, yeah.
Yes, the answer is yes, and the answer is partly no. I'll try and kind of give you both. So let's see. Go with I'm not going to do much with it, but I just pull it up so we can have something to kind of look at. So if we have, so this has got the time delay. We had another one that had a time delay. This will do though. I think you were actually my answer to your question in any case is, which I think is maybe what you were getting at. It's like, is the when we're using this kind of thing to model epilepsy, is it F or is it something more like well, Y? Sorry, what would it be? Our state variable be, yeah, Q. So we don't usually, the delay and the activation function aren't the things that are the epileptogenic parts of the system. The epileptogenic parts of the system is the coming from the dynamics of the neurons, and that depends on the kind of regime that they're in, but also often it's like where they are in relation to certain bifurcations within their like parameter space. And the thing that kind of nudges them towards, say, moving into the high firing, what we call epileptogenic phase, is in some cases that's thought of as like crossing a stability boundary. because it is like unstable, kind of high firing that you get in epilepsy. It's kind of quashed by metabolic kind of feedback at the end of the day. But it could be the case that the only thing that stops epileptic seizures kind of completely exploding is like a resource depletion. But in any case, all of those considerations, like what is it that's moving me into a seizing regime? What is it that about that being here versus here, or how it spreads from here to here? The way that that's, as far as I understand anyway, like the way that that is kind of expressed in the models is the tuning of the kind of local dynamics in relation to their kind of seizure versus non-seizure state, not really the activation functions or even the time delays. But the other thing that could contribute is the connectivity matrix structure, in the sense that like there's some, you might have a more seizure prone connectome, more seizure prone wiring structure in some people than others. More dense would be the kind of thing you would think give to more seizure prone. I'm not actually sure what the bottom line is on that. It's like whether people think that there's obvious things you can see when you look at the white matter and connection structure of epileptic patients versus people who don't have epilepsy. Do they kind of look? I'm not sure that that's obviously the case, but at least in principle, right? Kind of theoretically, like a dense connectivity would be even just intuitively more likely to give you a kind of exploding activity than something that's sparse. So there's and then you can break it down to certain connections as well. So if you think about propagation, and this is, I think, a kind of applied example I didn't have here, but I will talk about in a later lecture. Like there's certain points where you have seizure focus here, have the rest of the brain here, and activity kind of spreads through the seizure focus into this, what we call kind of propagation zone that isn't, it's not kind of independently moving into a seizure, but it will be nudged into a seizure by an adjacent bit of brain area that is seizing, and then it kind of distributes it out globally. So those parts of the wiring structure, they're kind of high, I'd say. There's a graph theory term for this that I'm forgetting right now, but the kind of edge version of node degree. It's high centrality. It's high kind of between the centrality parts of your connectivity matrix. Those would be very important to kind of tone down in terms of suppressing seizure spread, because they kind of allow inputs from seizing zones to be kind of distributed out. So that's something that's part of the wiring structure that could be kind of promoted, or from a kind of clinical point of view could be surgically suppressed, right? That's not the dynamics as such, it's the wiring, but it's not the activation function really, and it's not the delay either. It's more like the weights matrix.
Right, so it's like, well, I guess in modeling terms, you'd have to give some initial conditions or assumptions to the connectivity, like I'm kind of understanding it as you have activation, and then upon that activation, we also have a time component, so that's not instantaneous, and that energy, that like neural energy, kind of can flow throughout neurons, but the actual neurons itself, the actual like geography of it, that has to be assumed in a certain way, and then we potentially could say people with epilepsy have that geography looks oddly hyper-connected, such that it creates some sort of storm in their brain when you let that energy loose.
Yeah, I think that's certainly kind of right, in terms of that's part of how people, I think, largely intuitively approach this topic, but also, that's one of the dimensions of like current scientific thinking. What I can definitely say is kind of live in the sense of this has got to the level of clinical trials, which is quite a big deal for computational neuroscience, is using this kind of understanding, broadly speaking, neural fields, but more like activation spreading in networks, to guide surgical decision making in terms of what to cut. Now, whether I kind of have to say this, I'm not sure whether the flip side of that being we can concretely say people with epilepsy have more epileptogenic connectivity structure. Is that an experimental observation that's like known to be the case? I'm not actually sure about that, but I mean, there's epilepsy is such a kind of complex condition because you have there's similarly to the general scenario I've been painting about there being structure at lots of levels, like the epileptogenicity is also something that's kind of a multi-level phenomenon because at the base level it's neurons firing, right? But at the most important kind of clinical level, it's most of the brain having a seizure, which is like many neurons firing and there being a certain like spreading phenomenon to it. But it has to propagate up from neurons, but the level of the kind of pathology doesn't necessarily need to be, well, at the most kind of atomic unit level, right? It could be like the wiring structure leads to neurons firing and spreading, but the thing that's kind of wrong, if you like, is something that's in the middle rather than at the bottom or at the top. And it's not my field, so I shouldn't kind of go too far in it, but I don't think that the answer is clear on like what the right way to understand the kind of fully unfolded pathology of epilepsy is, but it's clearly a combination of those things.
I'm trying to link back the idea of like activation, it's just like energy flow throughout a field.
Yeah.
I was thinking like, okay, I don't know if it's true, but could I electrically induce a seizure onto a person? And then if so, is that to say that maybe human brains have a general connectivity to them? And then what I could say is epilepsy is also induced from activation, how much like energy I'm putting in. And I'm now no longer trying to debate whether or not I'm changing the architecture and the connectivity matrix of a brain, but more so how much electricity I'm putting in. And those two debates, like, is it from the activation? Is it from the time dilation, just like fast, really strong electricity? Or is it from just a super connected brain that is like almost wired? I'll use the word loosely, poorly, such that you get a seizure? Or like, is it both kind of thing? That's what's like listening to the lecture and kind of getting to that.
So I as like a lot of things to kind of briefly comment on that you covered there, because firstly, like just at a practical level, we do research on brain stimulation, right? If we're in the lab and we stimulate someone's brain and they have a seizure, then we have to stop our experiment and cancel our study. We're not allowed to do that, fortunately. Right? It's a very strong kind of rule in place. So, and then practically, it's like the amplitude that you can apply, and it's kind of calibrated to the things that we can measure about how strong the response is. But broadly speaking, like I think in Toronto, certainly in some groups that I know, they've like over maybe a 20-year period, they have a seizure event in a TMS study every maybe 10 years or something, like one or two. It's like very rare, but when it happens, it's very serious. So we can't basically do that outside of the specific clinical case of you have someone with epilepsy and you're operating, or you have a pre-surgical kind of mapping where you have electrodes inserted into the brain. I'm not sure. I'm pretty sure there's like very stringent rules around what it is that they can do in terms of inducing seizures, but I think that would be the context where you would have an actual at least in humans, right, where you're actually inducing a seizure. In animal models, yeah, I mean, induce a seizure whenever you like. That's what they're there for, so to speak, like animal models of epilepsy. So, so you know, can't really do this in humans. Can do it maybe in patients, but certainly can't do it a lot. In any case, the other thing is, it's not just stimulate, activity, propagate. It's stimulate, epileptic activity, propagate, because stimulate, high activity, propagate, that's just neurons doing neurons. That's just the brain doing kind of what it does. You know, visual response or just a stimulus response kind of, and then say evolves over a couple 100 milliseconds, ripples through, then it kind of goes back to baseline. That's normal. The thing that would be characteristic of an epileptic seizure spread is like not neurons going up, like firing and then transiently firing and then stopping, is they go into a super high firing regime and they stay there for a long time. So they basically will be firing at kind of, I don't know, maybe 10 to 100 times their normal firing rate, certainly at least 10 times their normal firing rate, for periods of like tens of seconds, as opposed to briefly for like 5-10 milliseconds or 50 milliseconds, right? So yeah, you get the spreading, but the spreading in epilepsy isn't like ping and then it's just the kind of natural time constant of neurons firing and then decaying, and then that kind of moves through. It's more like you push a bunch of tissue into its unstable firing, a seizing regime, and then that pushes the next bit of tissue into its unstable seizing regime, and then you have that ripples through. But that's like it's kind of like a disease spread than a kind of than like activation pulse, because the seizing state is more persistent. I mean, it doesn't last forever. It lasts for like tens of seconds usually, and then something will like move some seizing tissue out of a seizing state, and it might be a kind of has run out of energy kind of thing, like a metabolic factor. But yeah, so it's a certain kind of propagation that is got a kind of long time constant to it. I brought us back to here just to say that I want to cover some of these things that we've just been talking about. Well, actually, we will hit them again. So we did some of the like fundamentals on neural fields today. They'll come back. So lots of these things will come back. Wilson-Cowan model will come back. Some of the stuff in the next lecture will come back in certainly lecture 6. So the things I've just been talking about, this kind of seizure propagation stuff, that's more here because this is whole brain modeling. And so we'll talk about this again, is what I'm getting at. And at the end here, so I've got some, this is kind of clinical application final lecture here. There might be some room for adding in epilepsy on this, so we might be able to revisit again there. But in any case, like we will return to the question of epilepsy. We'll also return to the question of neural fields in the sense that I talked about with the diagram with the fluorescent axon tracing, and I was also relates to delays. And I'll just use this diagram here to just spell it out. So what we were looking at here when we were talking about these one-dimensional, these kind of fronts and bumps and stuff, you think about them as like somewhere, yeah, maybe like roughly the size of the red cursor here. I've got a little diagram. There you go. It could be larger. It could be here, right? But then when we want to talk about some patch of tissue connected to some other patch of tissue, say this part of the brain over to this part of the brain, then we need to add a little bit more in, okay? So we'll revisit neural fields in the context of large-scale whole brain neural networks, and there'll be a kind of multi-connectivity structure where we have some local, some long range. So we're not done with neural and delays. So we're not done with neural fields. but we've had some of the kind of we've certainly got the basics down today. Okay, I'm done. I think I was on my conclusion slide before, but I'll leave it here for now, but open to questions from everyone. And then we'll take a break. And then Paz is going to run a tutorial. And we can talk a little bit at the end about the assignments. So any other questions on the lecture? Nothing burning? Okay. Well, thanks for your attention. We'll take a 10-minute break. So it's 4 o'clock on the dot right now. So we'll see you back at 4:10, and Parza will take the room and bring up your browsers and pay attention to the Zoom link for the link to the tutorial that he'll be going through.
There we go. Okay, perfect. So as I was saying, the introduction here, the novelty for this lecture is the introduction of the spatial element to it. So this was the Wilson-County equations that we went through before, but now we're going to go through the Amari model in this tutorial. And okay, so let's get started with the equation itself. So let's see what this is saying. We have a u variable dependent on x and t. So x is space, t is time. We get an eta kernel, w kernel, and 2 convolutions, and an activation function f, and again the state variable u, or the activity level of our neurons in this case. So we're gonna, so again, U represents the neural activity, so you can think of it as the membrane potential, or the voltage, and the neural response function is, again, if you remember from lecture 2, I think we had like a sigmoid function which translated our membrane voltage to firing rate. It's kind of the same idea here, and then over here, we can have it in 2 forms, either a sigmoid or a heaviside function. And I know I'm brushing over these, but I'll come back to this, and we can explore what they actually mean. And then we have a connectivity kernel. So the connectivity kernel here is what's actually adding a lot of the spatial dynamics, so specifically the center surround inhibition that John was talking about in the lecture. Okay. Let's go through a step-by-step construction of the field equations. All right, so let's assume that we have a field like so. This is our one-dimensional field of neurons, and we can have activity patterns over it. So here, let's just say we have 80 centimeters of cortex, one-dimensional, and there's an activity bumper activity in the middle. So you want to see how this interacts with the regions surrounding it and how they actually influence it back. So we'll start with this. We'll start with the activity, the initial state activity. So this initial state activity gets passed through an activation function. It's a neural activation function, which is essentially, you can think of it as translating into a firing rate. So over here. So this is our response function u, and this is what happens in the first bit of our, or the primary bit of our equation. We have our neural activity, and when we pass it through our response function, we can translate that into kind of like a firing rate, which is what actually gets communicated between the different neurons. And so, like for example, let me see if it's running. Yeah. So depending on the initial condition of our field, you can see how that translates into firing rate. This is a pretty straightforward operation, just translating that membrane potential into firing rates. We've seen this multiple times before in the other tutorials as well. So the second step is, so we were over here. We were just looking at this part right here. So the second step is the spatial convolution. So in the equation here, we actually have two different convolutions. This sign right here, the X, is a spatial convolution. The O here would be a temporal convolution. Right now, we're going to focus on the spatial convolution. So it's a spatial convolution of our firing rate within a spatial kernel. So what does that mean? Here, you can imagine that if you have neurons, the idea behind that is the neurons, the connectivity patterns in the cortex are both excitatory and inhibitory. Excitatory within their nearby neurons, and surrounding that, the connections become inhibitory. So they suppress the activity around them, and they amplify their activity immediately surrounding them. So this is what it would look like. This would be our connectivity kernel. So what we would do here, and for the spatial convolution step, is to convolve the activation, the firing rate pattern that we had within the connectivity kernel, with the spatial connectivity kernel. And that what that would lead to is this kind of green type activation pattern, or communication pattern rather. So let's see. And the way that we construct the kernel here is essentially, as John was saying, using two different forms, but here is exponential decay functions, two different exponential decay functions with slightly different variables. So we've got an excitatory one and an inhibitory one, and if we subtract them from each other, we kind of get this shape of like it going down and coming back up. So for example, if I change the contribution of the excitatory and inhibitory, you can see that the shape of the kernel is changing. There's also this gamma parameter, which is essentially the inhibition strength. It's a scalar multiplied by the inhibitory factor of the kernel. They can see that if I increase it, it will become more inhibitory, and the resulting firing pattern would become more inhibitory, more inhibition effect. So the intuition so far is that if you have neural activity, they translate into firing pattern, and the ones that are surrounding our region of interest, ones surrounding our point are becoming excitatory, and the ones a little bit farther away become inhibitory. And so that's we're 75% over with the equation. There's one extra step of the temporal convolution. So the temporal convolution is essentially how it evolves over time, how it plays out over time, which is, again, we kind of like seen similar concepts before. Here, this is the same. This is the synaptic kernel that we were talking about in lecture 2, if you guys remember. And so this is what we had from step 3. So this gets convolved with temporarily convolved with their synaptic kernel, and what it would lead to is a pattern that changes over time. So these traces are essentially telling you how this pattern is changing over time at different times. So you can kind of like see it decaying, and if you generate like an animation for it to actually get a sense of what's happening, let's see. So it's kind of becomes like that. So stop it. So this is our initial state. This is how we initialize the model, right? There was one bump in the center, and we want to see how this communicates with the surrounding region and what happens. So you can see that immediately, well, there's a decay effect because of the temporal decay, the convolution. But if you pay attention also, you can see that the surrounding region has become inhibited while the center region is more excitatory. So this is what's happening when we have like a perturbation, and this is how the field communicates spatially. Any questions so far? Scaling gamma would influence your connectivity kernel, which would influence the final results, but it would be, let's see. So it's changing the shape of the kernel. It doesn't change the shape of the synaptic kernel, but it would change the final output. So if I increase it, then I would expect it to be kind of like decay more faster, essentially. All right, so that was the convolution form. but you can also turn that the convolution form into an integral differential form. And the way that we're doing it here is by assuming that the synaptic kernel is an exponential decay function. So once we do that, it's just substituting eta with this part right here, and then writing it out, differentiating both sides with respect to time, and then we get form that's kind of more familiar. That there we get a value for the field as we can describe the field as like a dynamical system where you get the rate of change determined by the value itself plus the contributions from surrounding regions of activity. And if we just use this model to this form to simulate our data, it's essentially the same thing. We get the same thing. So instead of an animation here, you're seeing a heat map. So over here is time and over here is space. So essentially, you can think of it as this is a plane with a bump in the center, and this bump gradually decays to 0, and then your field goes back to a baseline. Couple of exercises for you guys. Now, so that was the equation, and that's basically the whole equation that's giving us this spatial communication. And within this equation, we can have different interesting patterns emerge because, you know, the point of defining the equation is to actually like observe, to model different interesting patterns with it. So to see what we can do with it, we can, depending on the variables that we have, depending on different existence criteria, we can have different stable structures. So one of the stable structures that we can have is what's called a stationary front. And essentially, you can think of it as one region is, it's a stable pattern that doesn't change over time. And part of the field is active and remains active, and part of the field is inactive and remains inactive. So the existence criteria is this, I can get into that if yeah, let me just show you first. So it's the color scheme isn't the greatest here, but essentially you can see that the state of the system evolves into a very stable pattern that doesn't change anymore. And you got regions where there's inhibition or inactivation at 0. And then there's regions of activity persisting over time. And the existence criteria is pretty straightforward, honestly. It just has, so it has to do with the synaptic response function. We could, in this case, we're using a step side, heavy side function, and the existence criteria is that the neurons or the states that are inactive should fall to the left side of our activation function to remain inactive, and the ones that are already active should fall to the right side of it so that they remain active. So it kind of like pushes either side to the extremes, and you get that stable pattern persisting. And the reason that it's kind of like a continuous form, like a sigmoid, even though we're using a heavy side, I think there was one of a question in the lecture, is because we're convolving this with a spatial kernel, and that spatial kernel gives us that smoothing effect that we don't see, like that's literal, like very sharp step. Stationary bumps. So that's one pattern. That's one stable pattern that we are seeing. But another stable pattern that we can get is a bump. So it's essentially like, you know, no activity and then bump out activity and then no activity again. So the existence criteria of that is pretty much based on the same intuition. One is that, I forgot to mention, one is that within our integral differential form, it's stable, so the rate of change is 0. So we can put the rate of change to 0. But in addition to that, it goes back to, again, the synaptic response function, where there has to be, the region in the middle has to fall into this kind of like active state, and the regions surrounding it have to fall over here. So that's what we're doing essentially here. So we're saying for x bigger than theta, or for values greater than theta, this is positive. sorry, when x falls between 0 and delta, so like the width of that of our bump, it has to be positive. It has to go into that positive aspect of the activation function, and otherwise it's 0 or it's negative. And again, when we simulate that, this is what it kind of looks like. So we start out with this blue trace right here, and we can see that it evolves into the stable form that is like a bump. So it's got a little inhibitions on the side, but it's a persistent bump that we're seeing here. And lastly, another stable pattern that we, quote unquote, stable pattern that we can get is a traveling wave. Now, the traveling wave wouldn't be stable because, you know, it's traveling, it's moving, but we can apply a trick to actually use the same principles as the stable wave, which is essentially to also have a moving reference frame, which we're assuming a reference frame is also moving. So it's like, you know, you're in a if you're in a car and you look outside your window and see other cars moving, to you they seem stationary. So it's essentially the same principle. You just change your reference frame to a moving one, and then you kind of like, yeah, you move things around, and essentially you can solve it similar to the previous steps. And what we're getting here, so I should note that within the numerical simulations, it's not exactly, it doesn't always exactly follow the math and the theory because of a lot of different factors, numerical instability, and the fact that what we're simulating is within a bounded region. But so essentially, this is what's happening. I'm initializing it in a particular state here. It's just 0, and then it kind of like goes up. But then based on this specific configuration that I have, a specific region I would get a bump in a specific region, and this region would kind of be slowly moving. Now, the speed of how it moves could actually change, but I can also show you a animation for it to get a better feel of what's going on. Takes a bit of time, but it's gonna be worth it. It's kind of cool. Any questions till it generates? So this is the blue trace is the initial condition. What's actually we initialize it with, and then see, whoop, and then it slowly starts moving. So we kind of like a kind of wave that's moving over time with the configuration that we had. So that's kind of cool. Yeah, so these are the different, I'm not sure, I don't think this is the only different patterns that you can get, but these are some interesting patterns, stable patterns that you can get based on the existence criteria that you define. Questions so far? Cool. This is still one-dimensional, yes. We're going to see examples of two-dimensional fields, but essentially it's the same principle. Instead of
Could I just imagine wave with the same kind of shape?
Exactly. So you can have in 2 dimensions, you can have bumps, you can have waves that extend this way, waves that extend that way, kind of like over, or like, you know, like platforms.
The choice of the activation function.
Activation functions and your synaptic connectivity kernel. So essentially it's really dependent on two factors, 2 kernels. One is a temporal kernel, which is the synaptic connectivity, and the other one is a spatial kernel, which gives you that center surround inhibition effect. And interesting. Right, if you consider the time delays. But if in the Amari model, even though in the Wilson-Cowan, if you remember, we had an excitatory neuron, set of excitatory neurons, and also an inhibitory neurons. But here we only have one set of one type of neurons, quote unquote. The inhibition and activation is not really coming from a different variable, inhibitory or excitatory. It's just coming from that synaptic kernel that we define, or sorry, temporal kernel that we define. And so yeah, so now I'm going to show you a couple of applications of neural fields. One is
there's excitation activation everywhere.
right.
No, there aren't those 2 populations. They're not separate.
Right. Right. So applications. The first application, and this is like, how we can use this model to apply it to different observations that we see, and matching it and kind of like assuming that, or like trying to describe the phenomena using our theory and intuitions that we've built with the theory. So here, another element, I just want to point out that another element is being added, which is external input to our field. So far what we've been talking about is just the field itself. We initialize it at a specific condition, and we see how it evolves. But we can also have inputs to our field. And so in our examples, the input is also being incorporated. One example is visual attention. So what actually like it captures attention in a visual field. So in this case, it would be some sort of like you can think of it as like a salience. If the stimulus is strong enough, then your attention will pop onto it. So what's happening here is that imagine this is your input stimulus to your retina, or your kind of like perception. And in the neural field, initially it's 0 because this is how it's initialized, and this gets added onto it, right? But after again, because of that kind of like filtering effect, it will pop onto if the stimulus is strong enough, then it will kind of like get an activation, and like your attention will be focused on the region of stimulation. Let's see what else we have here. This is animation of the same thing. The other thing, another application, is working memory. So John briefly touched upon this. So the working memory, what we mean here is that the stimulus persists even when the input is removed. So when you're kind of like, you hear something, you can hold it in your mind even though the verbal stimulus is gone. So what that would mean. There we go. Okay. So here, essentially, the blue is our neural field in the initial condition, and we add a stimulus about midway through. So, and the thing is, if we remove the stimulus again, the pattern still remains. So here, we've added so this is our 0, there's 0 condition, and when we add the inputs, it kind of like falls into matching the shape of that input. But when we remove that input as well, you can see that the neural configuration still persists and captures the same shape of the input that was given. And so this was going to be like a model of working memory or short-term memory in general. There's other applications of neural fields for EEG modeling. You can think of it as like a field over your scalp or your brain. Binocular rivalry, which John talked about. The activation of one region would inhibit the activation of one eye would inhibit the other eye, similar to that boundary regions in the kernel that we went through. There's visual hallucinations, which is interesting because in the case of visual hallucinations, what it's saying is that through, for example, through drugs, the configuration of your field changes so that, in a very hand-wavy explanation, so you would get self-forming patterns emerging, and they, because of this dynamic that I told you about, this stable configurations forming, you would get different percepts of different items in your visual field. Let's see, what else? Motion perception and epileptic seizures, John talked about motion perception as well. When the movement is actually, when your attention is tracking the movement of the stimulus as well, which would emerge out of the field equations as well. So, I mean, that was kind of a fast run through of the tutorial, not gonna lie. I thought it would take much longer. But yeah, so they just give you a summary of what we very quickly went through over the tutorial was the neural field equations. We broke it down to understand how it's constructed and what each part of it means, the other form of it, the integral differential form of it. And then we looked at the different spatial patterns that we can get out of the equations, namely the stationary front, stationary bump, and the traveling waves. We didn't cover the time delays in this tutorial, but we also looked at the different applications, visual attention and working memory specifically in this case. I see there's a question. Hi, Professor. I'm not a professor, but thank you anyway. Yeah. Could you go over the firing rates part? I missed that one. Yeah, of course. So this is similar to, I think, what we had in lecture 2. We're saying that we have membrane, but our variable is, in this case, representing membrane voltage or membrane potential. And the way that the neurons would communicate, I'll tell you about, like, conceptually, the way that the neurons are communicating is through different firing rates. So binary firing rates, which gets or we get so let me just, there we go. So this is an operation that would transform our variable from being a membrane potential of voltage into a firing rate. So what this means is that when you can think of it this way: when it's below a certain amount, the neuron is not firing, but once it goes beyond a certain threshold, then it starts firing, and it's as you increase it, it starts firing more and more until it saturates at some point. And because we have a population, we can kind of like smooth this out to an approximate, either using a sigmoid, or if you want to be more precise, we can have a Heaviside function, something like this, which would be off if it's below threshold, the neuronal threshold, and on firing at full capacity if it's above threshold. So what this is essentially saying with the firing rate bit is that we're trying to convert this membrane potential into a firing rate, which then gets convolved by the spatial filter, which would then give us the what we need for the communication between different regions. Over here, yeah. So it's essentially it's a sigmoid function. It's a sigmoid operation or a Heaviside operation. Firing is the sending message. I mean signal.
Firing is like sending messages.
Yeah, it's how you would communicate. Yes.
Yeah, firing is the output. Then synaptic response is the integration.
Yeah. Yeah, that's a good question. So it's through my experience when putting this code together, the model is extremely sensitive to different parameters because there's other factors going on in the simulations. Because this is a numerical simulation, not necessarily an analytical solution based on the slides that we went through. So it really depends, and there's also like boundary conditions. Let me just show you. So if it's an infinite field, for example, let's see. This is supposed to be a bump, so this is supposed to be a wave which turns into a bump. Yeah. So you can, for example, if you have an infinite field, then you wouldn't necessarily be exposed to boundary conditions. But when you have boundaries, then it doesn't yeah, it kind of gets like ripple effect back onto the thing, and it would kind of like influence the stability.
The front is the interpretation. Yeah.
So there's in the equations we integrate over from negative infinity to positive infinity, but here we're saying from negative 200 to positive 200, and the fact that we do have that boundary might influence the stability of the wave.
time. That's your initial conditions, transient.
So it's supposed to be it's supposed to be a front, and the front itself is moving, so it becomes shorter and shorter. So,
Okay.
but here it's not necessarily front. It becomes turns into a bump.
It's supposed to be a front because did you do a burn-in for the simulation?
I didn't do a burn-in. Well, the burn-in is like what you're seeing here, but essentially the conditions to get a front is that the neurons, the neurons that are off should be on the left side of their activation function, and the ones that are on should be on the right side.
Yeah.
And there's a bit of a for the bump, it has to be that the other side of the field as well should be on the left side of the firing rates response function. But even if I don't explicitly state that, the stable pattern that emerges is a bump rather than a front.
Yeah.
Which is what we're seeing here.
So what I mean, my interpretation of what you've got here would be that you have a transient due to your initial conditions that then disappears because that's what transients do, and the activity resolves down to your stable bump.
Right.
But the interpretation of the first bit wouldn't be that it's a stable front, or even that it's a front per se. It's just the
Right.
Injection transient.
That's true. That's true. But we can also have a stable front. For example, this was a bump, but in this case, we do have a front. So even like we can have stable fronts as well. So in the traveling front, this would be a front. So it would be a bump if this also comes back down to 0. No, not the color scheme is a bit wonky, but if I change it, let's see. Min.
I mean, I think another part of the answer to this question is I'm afraid the bit that wasn't covered in the lecture, which is the stability of the weight.
Right.
So if we cover that, then we look at this Evans function methodology that defines a linear operator on the traveling wave that you can look for, you can evaluate stability, and then when the eigenvalues are stable, so condition stability, then the traveling wave persists, and then when it's unstable, then it basically disperses to its so that's something that it's like, I think it's not exactly a contraction from a front to a bump, but it is like a stable traveling wave versus an unstable traveling wave. But not all traveling waves will be like the same time.
Yep.
Before this course, the only time I've really seen or debated like activation functions is from the lens of machine learning, where they talk about it. It's like, oh, should I use sigmoid? Should I use ReLU so I don't have disappearing gradients?
Right.
So like the debate on which activation function to use was like more so based on like what would give me the best output for this algorithm kind of thing. In this case, though, is the debate on the activation function, is it supposed to mimic more so like real neurobiology? Is there like, do you define strength of firing based on frequency versus like voltage?
It depends on the model that you're using. If your model is using, for example, the firing rate to communicate within different populations or different regions, then you can have like that you would be using firing rate.
Yeah, like the step function where it's like 0 or 1, like a Dirac kind of whatever, or it's like kind of, I saw it in the slides before where it looked more like a ReLU, where it was like that.
Right.
Yeah, is that also just like a neurobiology question? Is that a thing? Is it do you define like strength of neural activity as like is there a minimum amount of voltage to activate an electron? Or do you like define strength as frequency? Like if it only takes X amount of voltage to activate a neuron, then do we just define strength to be a lot of those, like repetitively, not just like a really high voltage?
So there's answers to this, and they kind of break down quickly, right? So is there a minimum amount of current to induce a memory potential change that to induce firing? Yeah, it's the amount that moves it past its firing threshold, which is typically the 50 kilovolts, right? So there's a number that is different for different neurons, and it's kind of state dependent as well. But it's the firing threshold that where the cell is firing for us. Now that's something the inputs. It's not just that one input, but one cell has hundreds of inputs, thousands of inputs, right? So there's the you've got to kind of figure in at some level like how you're thinking about the aggregation of the inputs, and they also there's a spatial piece as well because they occur on the depth of the tree, and then their effect in terms of pushing the memory potential down. Kind of that's the linear cable equation part of the first lecture. So kind of trying to the neuron and affects some of this another part within the cell. There's a spatial part of that. So even just for one cell, right? Like, yes, you can define what the activation threshold is somewhat concretely, but how that relates to inputs kind of depends on a lot of things about the cell. Then you add that up to a population, and then there's other things that come in. You have like distribution neurons. Those all have maybe slightly different firing thresholds, and how do you define what the input is? Because are you injecting an input into each neuron in the same way? Well, of course not. You're just sticking it into the tissue. So there's like tissue propagation. So what that boils down to is like for any given experimental setup, there are parameters that will be relevant for the thing that you're looking at, talking about activating neurons and stuff in terms of, say, the kind of things that we work on, which you can speak to most, right? So inducing a evoked response with TMS, okay, then you need to be somewhere above 100% of the motor. There's like a kind of calibration step where you see what amplitude makes the finger twitch when you stimulate the motor cortex, and you know that that's making neurons fire because neurons have to fire to get that muscle response. So that's like the thing that we kind of calibrate things against. It's like when we know that there's some, we can say for sure that's some neurons fired because that's the only way we can get something going up there here from something you did over here. But that's quite specific to that experimental setup aside. So you have the particularities of the experimental preparation, and then say, how do you connect that to the neuron model that you have of that? And it's hard to do a very concrete one-to-one mapping at a granular level of like physical quantities. You end up saying there's a threshold. I'm going to say the threshold is the following, and there's a sigmoid maybe defining some activation function. I'm going to just essentially phenomenologically determine it because it's pretty hard to do it bottom up because there's just so many things that will contribute to it. If you want to do it both like membranes up to TMS, you just can't do it.
So like a paper might show, like it might suggest the result with different maybe like one is like a bump or one's like a front, and then you'll get different Evan equations, like different stability based on those initial assumptions, and then you might extrapolate further on. I guess I'm curious, like from a always from like a mathematician point of view, I always want to know which variables are we fixing? And what are we assuming right now, so I know how to
Right.
gauge it.
Yeah.
And then biology can oftentimes like present those for me where it's like, this is just a biological phenomenon, like take it to be true. So I guess that's what I'm curious about, like, for example, minimum threshold firing rate.
Right.
You know, that could have like a gradient over the brain, as you're saying, like change different amounts, but I guess in this case, yeah, so if we were to take the in this case, we choose which like activation function.
For example, you can change the theta, which is the center of this activation function. You can, move it left or right to say how excitable on average are the neurons. So for one example, for one person, it might be that, their neurons are super excitable. So they will be activated at a lower threshold. So in that case, your activation function would be shifted a little bit to the left.
Right. This could look like you could extrapolate, like this is the one dimensional, you could have this model and it could like evolve over centimeters of your brain.
Right.
Like different, this part has more activation, less activation. This part looks more like this activation function or that activation function. Like it could be like a dynamical.
Yeah.
Right, that could be very tricky.
I mean, we have, you have like plasticity models and also like, habituation or desensitization models, which are, well, not implemented here, but essentially they're saying that the fixed parameters or the fixed constants that you're assuming themselves are changing over time, depending on like, activity of your brain or.
And that could just be like like you could say, I don't know, the activation between your like upper motor neurons and like getting someone to walk.
Right.
It's like you can see one person is like stronger than another kind of thing, and then maybe you want to find a way to like average that out or something. I get a lot of data from a lot of people or something to model.
Yeah, it would really depend on like, what exactly you're trying to like, what phenomena you're trying to model and what is your question. But like you would be justified to fix a lot of these parameters while trying to vary a lot, some a lot of other ones.
Usually the scenario I was describing where there's just lots of things that go into knowing what the kind of physical quantity is that obviously input that's inducing a response. That the neural model there, which is what we've been talking about here, usually that it's not always kind of fully articulated at the level of detailed neural components, because we're at this intermediate mesoscale where we have some kind of statistical quantities that don't kind of essentially describe they're describing something that's not an individual unit in the same way that like an ion channel is. However, the thing I was saying about when say how much stimulation you have to apply to induce a response from brain stimulation, or it could even be quite sort of light, right, or some like visual threshold thing. Usually the uncertainty there will just kind of pile in, we'll just put all of that into the calibration of the intensity of the input, because that's the thing that we don't really know. What we do know is at the model level that what we want the model to be saying about firing rates and things like population average memory potentials in this case, or so a lot of the kind of scaling that brings that we have no way of really summarizing down completely into kind of units. We'll just treat that as arbitrary units, and then we'll have some parameter that kind of just tweaks it into the range that then we plug in, and then we're talk about firing into that potentials again. So there's obviously some kind of parameters that move between our physical measurements that are not the kind of free or that we tune and just assume that they're kind of linking things in a way that's reasonable. We don't know what the exact number is.
John, we're getting to 5 o'clock, and I know you wanted to say something about the assignments.
Yep, good. So I'll do that and I'll wrap up. Any other final questions, brief ones before we do wrap up, guys? Speak now. No. Okay. So what I wanted to say is just to reiterate what we've said before. So there's a list of exercises in the note at the end of the tutorial, and I've posted the link. Sorry, I haven't. Yeah, I just posted the link for this tutorial in the page on the Moodle that has the, so to remind people, if you go into the Moodle and you go to the resources, there's a page that says tutorials, and on that page there's now 4 links, and those link to each of the notebooks that we've had accompanying each of the lectures. There's also the links to the slides and PDFs of the slides as well. So the assignment is to do all of the exercises for the first 4 lectures. They're not huge, okay? So
Right.
it's not like an epic amount of work. There's a kind of small set of essentially like extensions of the thing that's in each of the notebooks. What we want you to do, so in terms of the submission part of the assignment, is to copy download or copy the notebook that is linked to, and then just complete the bit at the bottom. So
Or in the middle, there might be in the middle as well.
in the middle, so it's clear where they are. Add comments, code, and just about narrative. I think if it's unclear to people what we mean by that, then feel free to ask, and we'll give some clear demonstrations. But I just want to leave it. I think it's probably get the gist. Like, we want you to take the notebooks, add content, follow the questions and the exercises that are described quite clearly in the text, and then indicate where you're doing it. And then that file is then your submission. Okay. Now for the submission of the assignments, there's 2 options to do this, and we're fine with either. One is you can email them to either myself or Parza. So if you want to do that, that's fine. And I will add this in the tutorial box on the Moodle, but it doesn't say this right now. But I mean, it says it has our emails in there. But what you can do for submitting the assignments when you've done them is to send an email that clearly says in the title, submission assignment, give us your name, and then it should have 4 files attached that are the 4 notebooks with your edits. Okay, so if you want to send them in by email, you can do that. The other option is you send them in through the Moodle, and there is functionality in there for accepting assignments, but I haven't tested it yet, so I can't right now tell you what the exact process is. But what I can say is, if someone wants to kind of click some buttons and do assignment through that, do a submission through that, and it seems straightforward, then go for it. I'll kind of we'll test that out as well in the next few days anyway. So if you want to be the guinea pig on the Moodle assignment, feel free. If you don't want to be the guinea pig on the Moodle assignment, then either email them or just hang tight until we have some follow-up on the Moodle submission option. And yeah, that's it. So assignment 1 is all of the
Notebooks.
components of the notebooks that are giving you questions and asking for some input from you from the first 4 lectures. Assignment 2 will be same thing for the next few. Any questions on that? No? Great. Okay. Thanks a lot. Thanks to Parza as well. Again.
Thank you guys. Thank you.
You guys next week.